Hello guys, welcome to Buddy Academy and this is me Uma here and I'm back today with a newer concept in biology. And before we go to our class, it's tip time. And here we go, tip number 3. Practicing from sample papers and previous year question papers is a must. So if you are thorough with the concepts, it is okay, it is good and you should be thorough with the concept. But you should know how the question comes in the exam and how the question paper is. So for that you have to have a knowledge of previous year papers and sample papers. So when you are preparing, you should also have an eye on that and you should be giving equal worth or equal weightage even for that. So let us move on to our class. Today we are going to begin a new unit, Genetics and Evolution. We are going to discuss about the chapter Principles of Inheritance and Variation. You know Genetics and Evolution contains 3 chapters, but this time since due to deleted portion it is having only 2 chapters, that is Principles of Inheritance and Variation and Molecular Basis of Inheritance. So you know this has the highest weightage out of all the units. So these 2 chapters alone will have a weightage of around 18 marks. So better don't concentrate only on important concepts. Try to concentrate and be thorough with all the concepts that are listed under these chapters. So let us move on to our class. So we are going to begin with genetics. So genetics is nothing but it is a study of heredity. So this subject will deal with inheritance as well as variation. So what is inheritance and variation? Inheritance is nothing but it is a process by which how you acquire your parental characters, and variation is nothing but how you vary from your parent in spite of acquiring that characters from your parent. So variation is obviously hidden in sexual reproduction. Because of sexual reproduction, variation is seen in many species. So before going into Mendel's law of inheritance, we will discuss about Johann Mendel, who is the father of genetics. So he was born in European country in a German speaking family and he was working in pea plant, that is with Pisum sativum, for 7 years. So these 7 years are very important to remember. He was working from the year 1856 till the year 1863. He was a mathematician but also a biologist. So even in spite of knowing maths, he was interested in biology and he applied those concepts of mathematical concepts in biology and interpreted many results. So after his death only many of his works were discovered, out of which we will just see eventually when his work was discovered and what did he conclude. So yes, Mendel conducted hybridization experiments on garden pea plant for 7 years. So remember this 7 years, you will also have 7 contrasting characters. So you can remember it like 7 years and 7 contrasting characters. So on observations on which he gave many laws of inheritance, which was called as Mendel's laws of inheritance. So Mendel investigated characters in garden pea plant that were manifested as 2 opposite traits. For example, tall and dwarf, yellow and green seeds, simply like that. So he conducted artificial pollination, that is cross pollination experiments using several true breeding lines. So true breeding lines are nothing but those plants would have undergone self-pollination for several generations and they will inherit a stable trait. For example, tall plants, if they are undergoing tall plants undergoing self-pollination for several generations, what will happen? Do you think will there be any variation? No, it would have that trait like a stable one. It would have passed on that stable tall trait throughout many generations. So he conducted cross-pollination and in spite of having the self-pollinating flowers, how did he conduct cross-pollination? So we have answer for this from your previous lesson itself using emasculation and bagging technique. So Mendel chose these characters from the plant Pisum sativum, that is garden pea plant. Then why did Mendel chose this garden pea plant is one important thing which you have learnt in your 10th standard itself. Again to remember, it is an annual plant. It will produce a various contrasting character and the lifespan of the plant is very low and it will also be self-pollinated or cross-pollinated. So this plant had many advantages. So for example, if Mendel would have chosen a tree, what would have happened? It will have a very longer lifespan, so that the experiment would have been hindered, or it would have been difficult to identify the particular character which is being passed on after generation. So these are the characters. So our seed shape, that is round and wrinkled, seed color yellow and green, seed coat color colored and white, pod shape inflated and constricted, pod color green and yellow, that is flower position axial and terminal, and stem length long and short or even dwarf. So before going into the chapter, you must know some concepts, that is definition for a gene and what is an allele. So we will study that, but then for you to understand, it is given in the beginning itself. So a gene is the basic physical and functional unit of heredity. So gene is nothing but a segment of DNA which is coding for a particular protein, or it is having a control over a particular character. So genes code for a pair of contrasting traits are known as alleles. Alleles are nothing but slightly different form of the same gene. For example, this A character is having 2 alleles. So this is A gene and it is having 2 alleles. You can see a capital A and small a. So homozygous are nothing but, I am telling you here itself, homozygous are if 2 characters, that is 2 genes that are present will be similar. If it is different, it is called as heterozygotes. So inheritance of one gene or monohybrid cross. So first we will know how one character is being inherited in a particular plant. For this, Mendel chose tall and dwarf plants. So what did he observe in the first generation is all plants were tall. So that first generation is called as filial 1 progeny or the F1 generation. It is also called as the first filial generation or the filial 1 progeny or the F1 plants. So he saw that all the plants that were obtained in the F1 generation were tall and none of them were dwarf. So when the same thing, so you can see this, after selfing the tall plants that were obtained in F1 generation, to his surprise, he could see some dwarf plants also present in the F2 generation in the ratio of 3:1. So if 3 plants were tall, 1 plant was dwarf. So now we are going to work out on this. So Mendel then self-pollinated F1 plants and found that the filial 2 generation, some of the offspring resembled the dwarf character which was not seen in F1 generation, and as I said, the ratio was 3:1. So if 3/4 of plants were tall, 1/4 was dwarf. So Mendel proposed that something was stably being passed down unchanged from the parent to offspring and through gametes. So over successive generations. So now we call them as genes, but Mendel called them as factors. So what did Mendel call genes as? He called them as factors, and he said that something is being passed down stably through generation, through gametes, that is from the parent to offspring, and now he named it as factors, and now we call that as genes. So they contain information that is required to express a particular trait in an organism. So now we are going to just do the cross for this. So let us take the alphabets for tall and dwarf plants. So for tall, I am taking it as T and T, that is 2 capital T, and for dwarf plants I am taking it as 2 small t. So why alphabetical representation is done is for easier understanding. We will be knowing why this capital T and small t are being taken, which is nothing but because of the dominant character. This will be studied under law of dominance. Because of the dominant character of this T, it is being taken as capital, and because of the recessive character of the small t, it is taken as smaller one. We are taking same alphabets like capital and small instead of taking T and D, because if we take like that, it will be difficult for us to understand. So these are the parental generation. I will name them as P, and now Mendel is crossing this and he will be getting TT plants in the F1 generation. So this square is a special square which is called as a Punnett square. So this was discovered by a British scientist called Punnett and named after him. So this square has many significance which we will be learning. Before that, we will just work out. So this is filial 1 generation, and again they are going to self cross this filial 1 generation. So now how will be the gamete formation? So this Punnett square, we can easily know the formation of gametes. This is one application of that. So now we are getting TT, capital T, small t, capital T, small t, and small t, small t. Just cross it like this, TT. Now T with capital T, again you will get that. So now this plant is again tall, this plant is also tall, this is also tall, whereas this plant is dwarf. So how did they tell that this plant is tall? So because of the dominant character of this capital T, this plant is tall plant, whereas the recessive character is not exhibited in that particular plant. So yes, this square is the Punnett square, which was developed by British geneticist Reginald C. Punnett. It is a graphical representation to calculate the probability of all genotype of the offspring. So again, I will be telling you what is the genotype of the offspring and what is exactly a Punnett square, a graphical representation to calculate the probability of all genotype. For that only they are using Punnett square. So now we are going into law of dominance. As Mendel found that the phenotype of F1 heterozygote to be exactly like tall parent in appearance. So yes, phenotype is nothing but the external appearance. And genotype is nothing but the genetic makeup. So this is the genetic appearance, how the genes are. And the other one is the phenotypic appearance. So as Mendel found the phenotype of F1 heterozygote to be exactly like capital T parent in appearance. So he proposed that in a pair of dissimilar factors, like for example, these are not similar. So what did he conclude? In a pair of dissimilar factor, one dominates the other and hence it is called as the dominant factor while the other is recessive. In this case, capital T for tallness is dominant over small t for dwarfness, that is recessive. Simply as you have learnt in your 10th standard, to conclude, the character which is being exhibited in the F1 generation is considered as the dominant character, whereas which is not expressed in the F1 generation and finally exhibited in the F2 generation is considered as a recessive character. So what did he conclude from this? Characters are controlled by discrete unit called factors and now we call them as genes. And how these genes occur? They occur only in pair. In a dissimilar pair of factor, one member of the pair dominates and that is called as dominant, whereas the other is recessive, that is not being expressed. So next law will be law of segregation. So when the tall and dwarf plant produce gametes by the process of meiosis, the alleles of the parental pair separate or segregate from each other and only one allele is transmitted to each gamete. So again when we come here, we can see these characters, right? So when these genes are segregating during meiosis, only one allele will be transmitted to each gamete. That is why we have given the gametes here. So before doing a Punnett square, we must also mention about the male and female gametes. So I am writing male and female gametes here. So yes, so we are having capital T and small t, right? So during the meiosis, what will happen? These will segregate, that is law of segregation, that is what they have mentioned here. This segregation of allele is a random process and there will be a 50% chance of gamete containing either of the allele and it is verified by the results of crossing. In this way, the gametes of TT plants have the allele capital T and the gametes of dwarf TT plants have a gamete of small t. So during fertilization, two alleles from the parent through the pollen and from the other parent then through the egg are united to produce zygotes that have one capital T and one small t. So then next will be the genotype and phenotype. What is a genotype and what is a phenotype? As we have already seen, the genotype is the genetic appearance and the phenotype is the physical outer appearance, how they look outside. So this genotypic ratio, so genotypic and phenotypic ratio are very important. Just these pictures are for your identification and in no way related to pea plant. So there are two ratios, genotypic ratio and phenotypic ratio. So genotypic ratio is nothing but when you see the cross. So what can you tell from this cross? So you can see that 3 plants are tall whereas 1 plant is dwarf. So first let us conclude the phenotypic ratio. I will just write it as phenotypic. So what can you conclude? 3 plants are tall whereas 1 plant is dwarf. What is the genotypic ratio here? So how the genes are differing? 2 plants are homozygous tall. So as I have already said, homozygous, similar alphabets. So here for your understanding we are telling similar alphabets will be present. So again heterozygotes, different things or different combinations are present. So you can see 1:2, that is 1 tall homozygous tall, 2 heterozygous tall and 1 homozygous dwarf are present in this. So yes, again genotypic ratio and phenotypic ratio are very important. Genotypic and phenotypic ratio of monohybrid cross are 1:2:1 and 3:1. So this was also given by the formula. So this 1/4, 1/2 and 1/4 ratio, that is the genotypic ratio, can also be mathematically condensable to form a binomial expression, that is ax plus by the whole square, which is nothing but ax plus by into ax plus by. So yes, Mendel self-pollinated F2 plants and found that dwarf F2 plants continue to generate dwarf plants in F3 and F4 generations even. So he concluded that the genotype of dwarfs were homozygous TT. So now it is test cross. We are going to discuss about test cross. So before using Punnett square, Mendel used this test cross to determine the genotype of the progeny. So how the genes are made up in the F1 tall plant, that is capital T small t plant. So this is an important thing. What is the significance of a test cross? It may come in your exam. So what he did is he crossed the F1 tall plant, that is capital T small t, with double recessive dwarf parent, that is with a dwarf parent progeny. Okay, so this is a dwarf parent. He is taking the F1 progeny and he is crossing it with a dwarf parent. So what should happen here? So according to Mendel, he should have obtained the same thing, like tall plants should have produced tall progeny since T is the dominant character, right? So but to his surprise, the ratio was 1:1. So if one tall plant was produced, one more dwarf plant was produced. So both the phenotypic, that is external appearance, and the genetic makeup was 1:1. So with this he concluded that one recessive gene should be present in this heterozygous tall plant. So that he said that this tall plant of F1 generation will also have an recessive trait. So based on his observations of monohybrid crosses, Mendel proposed 2 general rules to consolidate his understanding of inheritance in monohybrid cross. So today these rules are called as the principles or law of inheritance, that is law of dominance and law of segregation. So now we are going to discuss about incomplete dominance, where the F1 progeny will be an intermediate between the parents. So for example, this will be you can consider a dog flower, that is snapdragon or anthurium species as an example. So where if red and white colored flowers were crossed, an intermediate pink flower was produced in the F1 progeny. So this is the incomplete dominance in Mirabilis delaba. Mirabilis delaba is the 5 o'clock plant, and this also can be considered as an example. So what can you give as an example for incomplete dominance? You can give snapdragon, that is dog flower, and Mirabilis delaba. So yes, so what you can see here, that parent with red and white flowers. So if they are crossed, what they are getting in between? They are getting the pink flower in between. So you can tell that neither of them are dominant. So when they are continued to doing this in F2 generation, so selfing F1 generation, what they are getting is, they are getting 1:2:1 ratio, again both genotypic and phenotypic ratio, both geno and phenotypic ratio are 1:2:1. So this is incomplete dominance. The character will be an intermediate. So now, is this an exception for law of dominance? So what exactly is dominance? Why are some alleles dominant whereas some recessive? So every gene contains information to express a particular trait. So in diploid organism, there are 2 copies of each gene, that is a pair of alleles. So these need not always be similar as we find in homozygotes. It can be even different. Why it is different? That it has undergone, so changes it would have been undergone, which modifies the allele, modifies the information that particular allele will contain. So for example, let us take an gene that contains an information for producing an enzyme. Now there are 2 copies of gene, that is 2 allelic forms. So let us assume that the normal gene produces the normal enzyme that is required. For example, if a substrate S is going to be converted into product P, this normal gene will code for a normal enzyme which is going to do a normal function. It is going to convert S into P. So what will the modified allele do? It can be responsible for a less efficient enzyme or a non-functional enzyme or no enzyme at all. So here what will be the result? In the first case, whereas a normal or less efficient enzyme, the result would be the same as the unmodified allele. But when you take both these, a non-functional enzyme or no enzyme at all, what will happen? The entire thing itself will be hindered. So now what can you consider here is the recessive trait is seen due to a non-functional enzyme or no enzyme is being produced. That is why that trait is not being expressed. So next topic will be codominance. So till now we were discussing about F1 resembling either of its parents or it was an intermediate between both the parents. So now in codominance you can see the F1 will resemble both the parent. A classic example is the blood group AB alone. So you should remember only the blood group AB is an example for codominance. Generally, the plasma membrane of the red blood cells has sugar polymers that protrude from its surface and the kind of sugar is controlled by the gene I. So this gene I has 3 alleles. One is IA and IB and IO or even small I. So this IA and IB produce slightly different form of sugars, while the small I or IO does not produce any kind of sugar. So because human beings are diploid organism, each person possesses any 2 of the 3 I gene alleles. So you can know that we are diploid organism, so we will be possessing 2 alleles, so that is either IA, IB or IO. So these 3 can come in 6 combinations. So you can see this. Yes. So these IA and IA combines, it will be A blood group, and if IA and IB combines, it is an AB blood group. So this is the codominance. 2 characters are being expressed here. So this can give a combination of around 7 forms. So you can see here IA and small I is present, again blood group A. IA, IB is present, again blood group AB. So these can give a combination of around 6 blood groups. So you can see A, AB, A, again AB, BB and O. If IB and I naught, that is IO are present, again the blood group is going to be B, and if IO are present or 2 small i's are present, it is going to be the blood group O. So what can you conclude from this? But when IA and IB are present together, they both express their own type of sugar, and this is because of codominance. One more classical example you can draw is the coat color of cattle. When red and white color cattle were crossed, roan color cattle was produced, like it was having a red background on that white patches of cattle were seen. So this is the F1 generation. Again, when you take the F2 generation, what was seen is, so you can know now it will be like R again, which is red, and RW, I will just put as RW, RW, and again WW. So 1:2:1 ratio. So these RW are the roan colored cow. That is, it will have a red background and it will have a white patches on that. So do you realize that the example of ABO blood grouping also provides example for multiple allelism. So what is multiple allelism is more than 2 genes, that is alleles, are controlling or governing a particular or single character, that is called as multiple allelism. So you should be careful that the ABO blood grouping, so blood grouping ABO is an example for multiple allelism. Whereas when you take only blood group AB, it is for codominance. So only blood group B will come for codominance, whereas AB will come for multiple allelism. So there are more than 2, that is 3 alleles, more than 2, it may be even 4, 5 or 6. If it is more than 2 alleles governing a particular character, that is same character, it is called as multiple allelism. So occasionally a single gene product may have more than one effect. So here one more example is also given for, that is for start synthesis in pea seed. So it is controlled by gen. For this is an example of single gene product having more than one effect. So there are 2 alleles here, one is capital B and the other one is small b. When it is capital B, the grain size is very large and it has more amount of starch, whereas if it is small b, the seeds are wrinkled. So but what happens here is, if it is an intermediate size, so for example, if it is like this, one capital B and one small b, what will be the result is the seeds were of medium sized and it was round, it was not wrinkled. So you can see here these are BB, whereas this one are capital B and small d. So you can again cite this an example for incomplete dominance. Therefore, dominance is not an autonomous feature of a gene or gene product that it has information for. It depends as much on the gene product and the production of particular phenotype. So from this product, as it does not have the particular phenotype, we chose to examine in case more than one phenotype is influenced by the same gene. So we have seen how one character is being inherited. So now we are going to see the inheritance of 2 genes together, and that is generally called as an dihybrid cross. So here Mendel is taking 2 characters, which is nothing but the seed color and the shape of the seed. So one is the round yellow, which is dominant over the wrinkled green. So seed color and seed shape. So here round seed and wrinkled seed, yellow color and green color. So you know which is dominant here is going to be expressed. So it is the round yellow character which is going to be expressed with a genotype of capital R small r and capital Y small y. So again, when this F1, that is RRYY, capital R small r, capital Y small y, then capital R small r, capital Y small y is going to be cross, that is F1 is self cross. How many combinations are obtained in F2 generation? So these combinations can be given as 9:3:3:1, which is nothing but 9 were round yellow. So 3 were wrinkled yellow. So you can see new combination is obtained here, and that is called as recombination. 3 were round green. So round seeds with green color and wrinkled seeds with yellow color was produced. This one is the wrinkled green. So you can see how many different combinations are produced, and when Mendel self-hybridized F1 plants, he found that 3/4 had the yellow seeds with 1/4 had green. So the yellow-green color segregated in a ratio of 3:1, just like in a ratio of monohybrid cross. So here the ratio of yellow and green seeds were 3:1. So what is the phenotypic ratio of dihybrid cross? It will be 9:3:3:1, whereas the genotypic ratio will be 1:2:1:2:4:2:1:2:1. So you can remember 1, 2, 1, 2 and 4, again just ulta 2, 1, 2, 1, that's it. 1, 2, 1, 2, 4, then 2, 1, 2, 1. So this gave to a conclusion of law of independent assortment. So what here Mendel is stating is, when two pairs of contrasting traits are combined in a hybrid, segregation of one pair of character is independent of other pair of character. So what is he concluding from this? So we saw round and yellow color seed. This round and yellow feature of character segregation is independent of each other. That is, it can independent of each other. That is, this can be received by one pair of, that is your gamete, and this can be received by other gamete. So this can also form a combination with wrinkled, or this can also form a combination with green colored seeds. So yes, so here what Punnett square is used for, it can be effectively used to understand the independent segregation of two pairs of genes during meiosis. So you can see here, using this Punnett square only, we have seen the gamete formation. So now I will show you gamete formation, how to write that. This R can combine with this capital Y, or it can also combine with small y. So you can see two different combination, that is, but the parental combination was this one. But what they are telling in law of independent assortment is, segregation of this character, that is this yellow character, is independent of that round character. That is what they are telling in law of independent assortment. And what is the probability of that? It will be 50%. So if 50% of R and 50% of small r is independent from the segregation of 50% of capital Y and 50% of small y. So next will be chromosomal theory of inheritance. This is nothing but, so just we will be having unblusher. So we told that Mendel published his work in 1865, but for several years it remained unrecognized. Why? Because the communication was not easy in those period. So continuous variation were seen in sexual reproduction. Thirdly, as I said, Mendel is a mathematician. He used that approach and techniques in biology, with which biologists at that time did not accept. So three people, Hugo de Vries, Correns and von Teschmark, independently rediscovered the Mendel's results and inheritance of characters. So by this time, that is in 1900, due to advancements in microscopy that were taking place, scientists were able to observe carefully the cell division, so that how the chromosomes are being separated. So the discovery of structures in the nucleus that appear to be double and divide before the cell division, and these were called as chromosome. So by the year 1902, the chromosome movement during meiosis had been worked out. So this is the chromosomal movement during meiosis. So from all these conclusions, Sutton and Boveri noted that, and they proposed the chromosomal theory of inheritance. So Sutton and Boveri. So they said that the behavior of chromosome was parallel to the behavior of genes, that is segregation during meiosis, and the behavior, what they mention here is, whatever is being affected to the chromosome, it will also have an effect on gene and vice versa. So recall that you have studied the behavior of chromosome during meiosis, that is mitosis, equational division, and during meiosis, reduction, division. So when chromosome numbers are getting reduced, the number of genes also will get reduced. The important things to remember are that chromosomes as well as genes occur in pairs. The two alleles of gene pair are located on the homologous sites on the homologous chromosome. So 2 points are very important to remember. What are they? These are called as the chromosomal theory of inheritance, and who proposed that? Sutton and Boveri. Behavior of chromosome was parallel to the behavior of genes. Chromosome as well as gene will occur in pair. During anaphase of meiosis 1, two chromosome pair can align at the metaphase plate independent of each other. So you can see here how the germ cells are produced after meiosis 1 and meiosis 2. So you can see here again. So Sutton and Boveri argued that pairing and separation of a pair of chromosome would lead to the segregation of the pair of factors that they carried. So again, this is also an important point to remember. So they gave only the theory of inheritance, but the experimental verification was given by T.H. Morgan, that is Thomas Hunt Morgan. So he worked on Drosophila melanogaster, which is nothing but a fruit fly. So yes, he worked on Drosophila melanogaster, and Thomas Hunt Morgan worked on these because it could be easily grown in the synthetic medium that is available in the laboratory. They complete their life cycle within 2 weeks and single mating could produce a large number of offspring. So also there were clear differentiation between the male and female. Also it has many type of variation which can be seen with low powered microscope. So next will be linkage and recombination. Out of his observation, Morgan gave linkage. So Morgan carried out several dihybrid crosses in Drosophila to study the genes that were sex linked, that is genes that were linked on the sex chromosome. The crosses were similar to dihybrid crosses carried out by Mendel in peas. So what did he use? He used yellow-bodied white-eyed females to brown-bodied red-eyed males and intercrossed their F1 progeny. He observed that the two genes did not segregate independently of each other and the F2 ratio deviated very significantly from the 9:3:3:1 ratio which was expected. What did he find is the characters did not segregate independently. That is why he gave this linkage concept. Morgan and his group knew that these genes were located on the X chromosome and saw quickly that when two genes in a dihybrid cross are situated on the same chromosome, the proportion of the parental gene combination were much higher than the non-parental type. What they are concluding here is, for example, if this is a chromosome. So here if two genes are located very close to each other on a particular single chromosome, what is being expected? The segregation of these characters will not occur. They will be inheriting together for generations. Segregation may occur but it is very rare. So Morgan attributed this due to physical association or linkage of genes and coined the term linkage to describe the physical association of genes or chromosome, then the term recombination to describe the generation of non-parental gene combination. If it is a new combination, he called them as recombination, and if two genes are linked together, they will not segregate. It is highly rare for them to segregate, and for this he coined the term linkage. So he also found that when genes were grouped on the same chromosome, some genes were tightly linked, that is they show a very low percentage of recombination, that is the same parental combination will continue throughout many generations, whereas if it is loosely linked, they show many recombination. So for example, he found that genes with white and yellow were tightly linked and show only 1.3% of recombination, whereas white and miniature wing showed 37.2%. So what did he conclude from this? This is because of linkage. Low percent of recombination is because of the linkage of genes. So you can see linked gene are very close together, and even after crossover you can see small g and small a are together, capital G and capital A are together. So you can see here again same thing. So recombination do form but not very often. It is very rare. So this is crossing over. You can see this. If the genes are located very far from each other, after crossing over, this capital A is also having small b, this small a is also having small b. That is why the crossing over probability will be increased. That is new combination, not crossing over. A new recombinant probability will increase if the genes are present far away in a chromosome. So if they are present very close, that is if they are linked, there is very less chance of recombination. So there are two types of linkage. One is complete linkage and the other one is incomplete linkage. So complete linkage, as you know, it is a phenomenon where the parental combination will be appearing together for more than two or more generation in a continuous and regular manner. So here incomplete, they are widely located and they have chances of separation during crossing over, and that is called as incompletely linked gene, and the phenomenon of their inheritance is called as incomplete linkage. So next will be genetic mapping. This is a smaller concept which can be asked for a 1 mark. Alfred Sutherland used the frequency of recombination. So the principle is very important. So with which he did genetic mapping is he chose the frequency of recombination between the gene pairs on the same chromosome as a measure of distance between gene and map their position on the chromosome. So this is very important for you to remember. What did he do? He used the frequency of recombination between gene pairs on the same chromosome as what? As a measure of distance between genes, and he mapped their position on the chromosome. So today genetic map is extensively used as a starting point in sequencing of whole genomes, as was done in the case of human genome sequencing process. So who did this? And principle is very important, and one application you can study. So next will be polygenic inheritance. So polygenic inheritance is nothing but the number of dominant genes present will govern the strength of that particular character. For example, in human beings there are a wide range of heights and there are wide range of colors, right? So here when you take the pea plant, not like pea plant, similar to pea plant, in human beings only tall and dwarf human beings are not present, right? So there are a range of heights in between. So one more easily understandable example is the skin color of human beings. So for example, let us assume that 3 genes A, B and C control the skin color of human beings. Like for example, we are going to talk here about the power of the dominant genes, right? So for example, A, B, C dominant forms are responsible for the most darkest skin color, whereas all 3, if it is recessive, it will be representing the most lightest skin color. So for example, yeah, AA, BB, CC will have the darkest skin color, that is capital ones, whereas the smaller ones AA, BB, CC will have the lightest skin color. And you can see the various dominant genes present here. So for example, there can be many combinations of these genotype, right? For example, now we are going to talk about the number of dominant genes and the power they exhibit. That is what we are telling it as polygenic inheritance. So if all are dominant, all genes present are dominant, the person will be extremely dark. For example, so yes, the number of dominant genes here are 6, and secondly there should be 5 dominant genes. It is not that only this combination is existing, it can even be capital AA, capital B, small b and capital C, or sometimes even this can be recessive instead of that. So the number of dominant genes should be 5, they will be very dark. So you can see when this part is decreasing, the power of the character is also decreasing. That is what we tell it as polygenic inheritance. So you can see dark, that is 4 dominant gene, an intermediate person with 3 dominant gene, and then light person with 2 dominant gene, very light person with 1 dominant gene, and extremely light person is having all the genes as recessive ones. So next is pleiotropy. So here there are some instances that a single gene can exhibit multiple phenotypic expressions. So we are going to take an example as a disease phenylketonuria, which is characterized by the absence of the enzyme or inability of the body to convert phenylalanine into tyrosine because of the absence of the enzyme phenylalanine hydroxylase. So this disease is caused by mutation in the gene that codes for the enzyme phenylalanine hydroxylase. So this is a single gene mutation, but it will have various type of effects. So what are the effects? The person will have lighter skin, hair and eyes than brothers or sisters without the disease. Other symptoms will include eczema, recurrent vomiting, jerking movements in arms and legs, tremors, mood disorders or microcephaly. So next topic which we are going to discuss will be sex determination. So first who did that was Henking. So he determined sex in some insects. So Henking in the year 1891 could trace a specific nuclear structure all through spermatogenesis in a few insects, and it was observed by him that 50% of the sperm received the structure after spermatogenesis, whereas the other 50% of sperm did not receive it. So Henking gave this name as a X body. So he named that particular structure as X body, but he could not explain its significance. So further investigation by other scientists led to the conclusion that X body was in fact the X chromosome. So after the discovery of microscope and all those things, so it was also observed that in larger number of insects, the mechanism of sex determination will be of XO type. That is, all the X bear an additional X chromosome. For example, the female will have 2 X chromosome, whereas the male will have the chromosome type of XO. That is, they will only have 1 X chromosome. So on the other hand, some of the sperms bear the X chromosome, whereas the others do not. Eggs fertilized by sperm bearing an X chromosome would become female, and those fertilized by sperm that do not have an X chromosome become male. So here what can you conclude? The male will have less chromosome number when compared to female. So here, due to the involvement of X chromosome in the determination of sex, it was designated to be the sex chromosome, and the rest of the chromosomes were named as autosome. So what is a classical example for this is grasshopper. Grasshopper is a typical example for XO type of sex determination, in which males will only have 1 X chromosome besides the autosome, and apart from that, female will have a pair of X chromosome, that is 2X chromosome. So in human beings, as you have known from previous lessons, the sex determination is of XXXY type. So in many mammals, this XXXY pattern of sex determination is followed, where the females will be mostly in human beings. When you take, the females are homogametic, that is, they contain 2 X chromosome, and the males are heterogametes containing 1 X and 1 Y chromosome. So in Drosophila also, these species contain X and Y chromosome, but the sex is determined by a mechanism called as GENIC balance theory. So here, how the GENIC balance theory works out is, it is done by an ratio. It is nothing but number of X chromosomes divided by the number of sets of autosomes that are present. For example, if there are 3 X chromosomes, generally the female will have 2 X and male will have X and Y. If there are any defects or something that is present, so normally what happens is these ratios are produced. For a normal female to be produced, the ratio will be 1, that is 1. And for normal male to be produced, the ratio will be 0.5. So if others are being produced, so 0.66 is for the intersex. So you can remember this. This is also an important thing in your NEET exams. So you can remember this. 1 is for female, 0.5 is for male, and 0.566 is for intersex. Sometimes even 6, 7 can be coming. So have an eye on that. So these are meta male or super male, super females or super male. So again, normal female, if the ratio is 1, all are normal female. So this is the XY type of sex determination in human beings, which we have already seen. So in the above description, you have studied about sex determining mechanism, that is XO type and XY type. But in both the cases, male produce 2 different type of gametes, either without X chromosome or with X and Y chromosome. Such type is called as male heterogamy, as I said. So now we are going to see about female heterogamy, which is seen in ZW type of sex determination. So here AA is given because for the pair of autosomes. So here in birds, the females are heterogametic, where the males are homogametic. You can see again the same mechanism is being followed. A is just for autosomes. You need not confuse with that. Again, ZZ and ZW. If again Z with Z crossing, it is male. Z with W crossing, it is female. So again, pigeon, so this is in hen and this is in pigeon. So same mechanism is also being followed here. Just for giving you an example, I have taken 2 birds. So here what type of thing is produced here? So here the male is homogametic, and here what is produced? It is the female heterogamy. So if they are asking any difference between the male and human beings and bird sex determination, you can add these points also. So again, sex determination in human beings, same thing which you have already seen, father and mother. So here father is having, here it is male heterogamy. So one more important thing is sex determination in honeybees. So yes, sex determination in honeybees, so they have queen and drones. Generally 3 type of bees are present, 2 female, that is one will be queen and other will be worker bee. So both are female, only queen is able to reproduce, whereas the worker bee are infertile, whereas the drone will be father. So here in sex determination in honeybees, the female will contain complete set of chromosome, whereas the male will contain only half a set, that is haploid chromosome. So if there is no fertilization, there are some viable haploid drone developing from the gamete of the queen bee itself. So without fertilization, some haploid drones will develop. So they are without fertilization, they develop, and by the method of parthenogenesis. So if fertilization is occurring between the gametes, that is, for example, drone father is producing B gamete, A and B are coming, they will be developed either as it will be developed as a female bee, and their way of developing, method of developing will be different. That is, after the formation of sex, the method of developing will differ, and that is why queen bee or worker bee will be produced. So offspring formed from the union of a sperm and an egg develops as a female, whereas an unfertilized egg developed as a male. So whereas the unfertilized sperm will not develop because it will be a non-viable diploid drone. Only female can develop offsprings through parthenogenesis. So next topic will be mutation. So mutation will result in alteration of DNA sequences, consequently result in the changes of genotype or phenotype of an particular organism. So it will, if a single point, gene point mutation, so we will be studying in next chapter about all these types of mutation. If a small defect in gene occurs, what will happen? Mutation is a sudden inheritable change. So if small defect occurs in a particular gene, what will happen is it will be affecting the entire character. So how this mutation can be studied? It can be studied to a chart called pedigree analysis. It will show the record of inheritance of certain traits over 2 or more ancestral generation in the form of a family tree diagram. So these are some symbols that are being used in a pedigree chart or pedigree analysis. The square one is for male and a round is for female, and a diamond shaped one will be sex unspecified. So if they are shaded, they are affected individuals, and if there is a small line, one line in between, it will be mating. If there are 2 lines in between, like it will be mating between relatives itself. So here the parents are having offsprings and they are showing that it is unaffected. Here the parents are having an affected offspring, and 5 number gives the 5 unaffected offspring. So here the idea that the disorders are inherited had been prevailing in human society since long. So what is helping in this? Pedigree analysis will be. In the pedigree analysis, the inheritance of a particular trait is represented in a family tree over generation. So human genetics, this is used as a strong tool to study the abnormality or the disease that are present in an individual. So next, these are the genetic disorders about which we will be discussing in the next class. They are classified into Mendelian and chromosomal disorder. We will be discussing in next class about this in detail. So Mendelian disorder are genetic disorder which are showing Mendelian pattern of inheritance, and these are also called as monogenic diseases. They are caused by a mutation of single gene. So with a single basic defect, it causes pathological consequences. So how can these be found? Again through pedigree analysis. So now these are the major characters that should be known for an pedigree analysis. By knowing this table, you can crack any type of pedigree analysis. So autosomal dominant. So diseases can either be autosomal or allosomal. Okay, so it can be autosomal or it can be sex linked. There can be a defect present in the sex chromosome. When we are learning about genetic disorder, these things will be very clear to you. That is, autosomal dominant do not skip generation. Affected parents can have unaffected children, whereas autosomal recessive skips generation. Unaffected parents can have affected children. You just can know the difference between them. Affected parents and affected children, unaffected parents, affected children. So sex linked dominant never transfers from father to son. Sex linked, if it is a dominant, it never transfers from father to son. All daughters of affected father will be affected. Example, vitamin D resistant rickets. So then sex linked recessive, here males are more affected, will be transferred in a crisscross pattern. For example, mother to son, father to daughter in a crisscross manner. Example, hemophilia. So now let us crack 2 pedigree charts. You can see these are not skipping any generation, and affected parents can have unaffected children. So you can conclude this as autosomal dominant. So next pedigree chart will be, you can see unaffected parents, that is here it is skipping a generation. Unaffected parents can have affected children. So you can also conclude that this is skipping generation because both the parents are not affected, right? So this can be written as autosomal recessive. So sex link we will be discussing in the next class. So before that we will just do some previous year question paper analysis. So F1 progeny bearing violet flowers and snapdragon plant bearing red flowers were self to produce their respective F2 progeny. So compare these phenotypes. So we are going to compare the F1 progeny of pea plant and a snapdragon plant. You should remember, pea plant it will be showing dominance, whereas this will show incomplete dominance. So you should compare the progeny and tell about dominance and incomplete dominance. Explain the sex determination in birds. How does this differ from human beings? Again, about the Punnett square they have asked, name the type of cross that would help the genotype of a pea plant bearing violet flowers. Again, if it will be a test cross. Compare in 3 ways the chromosomal theory of inheritance as proposed by Sutton and Boveri, that of experimental results on pea plant that is proposed by Mendel. Here you can compare both things, what they both told. They will carry factors, it will occur in, factors are things, they will be in pairs and they are inherited after generation. You can also tell the difference which Mendel didn't tell. The behavior of chromosomes and genes were parallel. They also explained about the chromosomes and their separation during meiosis, which Mendel didn't tell. You can mention those points. So explain linkage and recombination put forth by T.H. Morgan based on its experiment using melanogaster crossing experiment. You can tell that when T.H. Morgan crossed the yellow-eyed and white-eyed Drosophila, what happened? Here the parental combinations were more concurrent than the recombinant one. Why? Because it was closely linked. And then again they are asked the question about genetic mapping. You can mention the principle about that. And the final question is differentiate between incomplete and codominance, and you have to support your answer with example for each. So you have to tell about incomplete dominance and codominance. You should also give examples for them and you have to explain and compare them. Yes, so with this you can practice these things. You have to practice Punnett square for all the crosses, because then only you can easily identify the phenotypic and genotypic ratio. You should also know the pedigree analysis table which I have given, and you can crack various pedigree charts for you to be easy and thorough in the exam. And with this we are closing this class, and in next class we will just have a continuation of this lesson. So today we have discussed about law of dominance, law of segregation, law of independent assortment, incomplete dominance, codominance, multiple allelism, chromosomal theory of inheritance, linkage and recombination. We have also discussed about polygenic inheritance, sex determination, mutation and pedigree analysis. Followed by this, in next class we will discuss about genetic disorder, which is the only concept left in this chapter, and we will also begin with a new chapter, molecular basis of inheritance. Hope you have all understood my class, and before I leave, it's time for you to prepare. Schedule, prepare and score. Thank you. Bye-bye.
For more queries, please contact Buddy on 98408 44987 or visit buddy.life.